recording this because I've been putting them onto the YouTube channel for reference. So tonight is members find. So what we do is we are taking turns showing off the mushrooms we have found over the past week or so, and either suggesting IDs or asking for help with the IDs, and we'll do the best we can to try to figure it out. We take turns. So if you would like to, everyone is welcome to share. So if you have stuff you would like to share, go ahead and get in line by putting your name into the chat box, and I'll just call on people in the order that they sign up. We ask everyone to share the time. So to take, 5 minutes or so of the night for your personal observations, and limit yourself to like 3 to 5. As the mushroom season goes on, we definitely have to be cognizant of the time because we have a hard time fitting everyone in as more and more people are finding mushrooms. So we might as well get in the habit of sharing the time early. Anyway, I did have some people email me, and I see Hervé has some observations to share. So we might as well jump right into that. And if somebody could volunteer to type the names into the chat as we're discussing them, the names of the mushrooms, I think that always helps everyone. So why don't we go right into Hervé's stuff? You want to go, Hervé? And just a reminder to unmute yourself when you start sharing, because
Can you see my screen? Because I don't see it. Should be.
We do. I can see it. You might want to expand it a little bit.
Okay, can you see it?
Yes.
Okay, I found that recently. This is on the dead branch. They look like little nests, but actually when you look closely, you'll notice that they are not nests. They're like a jelly mushrooms. And what looks like a crown is actually the bark of the branch that have been lifted. So I think there are some yellow jelly mushrooms. They grew from under the bark.
Yeah, I know what that is. I'm trying to find the name for you. I've only ever seen that one time.
Yes, same here. I've found this. I forget the name, and it does burst through the bark. That is a correct assessment.
And you can see at the bottom here.
We found this at Bald Eagle when we were scouting. Do you remember? I forget the name of this thing. I think I put it on Mushroom Observer, and there were 1 or 2 false starts with the name. And I think John Plischke may have finally straightened us out on that one.
I remember this being on MO, not necessarily your observation, Dave, but somebody else posted this mushroom before. I think they gave it a name, but it escapes me.
The branch has about the size of my index.
Yeah, I'm looking in the ASCO book now to see if I can find it.
I have a posting of this on Mushroom Observer. I just can't remember what it's called.
Very interesting looking.
And you can see at the bottom here where I have my, the bark has been lifted, but there is no mushroom inside. So we can see that the crown is actually bark that have been lifted. You see what I mean?
Yes.
At the bottom, yes. It looks like there is a crown, but nothing in it.
Maybe an old one that's decayed away already.
Yes, maybe they are like mushroom from last fall.
No,
and most of it had disappeared.
No, these are definitely fresh. This is a spring mushroom.
You think they're fresh?
Definitely.
Yeah, some of them might be a little older, but the ones that we're viewing head on, those probably haven't been out that long.
Here we go. Cocomyces triangularis.
Yeah, there you go. All right, way to go, Luke.
I'll type that in there
Triangularis, yeah.
for everyone.
Is it because the bark looks like triangles?
That's a good question. Why is it called triangularis? That's a cool
piece-
find. I don't think that's seen very often.
The
Yeah.
little pieces of bark look triangular that come up.
Yeah, you see what I meant, yeah.
Mhm.
I've never seen it ever.
Mm.
Because first I thought it was part of the mushroom, you know. I thought the crown was, I thought there was some kind of nest mushroom.
Yeah, I don't know
And
why it's called that because the spores are needle shaped. Well, anyway, there you go. Cocomyces triangularis.
Okay, very good.
Nice find.
Yeah. Next one. Okay, I'm going to show the next one. I think this is chestnut blight, but I need some confirmation. This is an American chestnut. This is in Hilliard Woods, you know, in Rutgers Gardens. There are about 3 American chestnut. They generate like shoots every year, and then the shoots die. So they never get taller than like 4 feet. And I see some something orange growing, and I suspect that this is the blight, that the blight is still active. The blight from the 1940s, because Hilliard Woods used to be, that Hilliard Woods is an old growth forest, and the dominant tree used to be the American chestnut. So it was a disaster in the 40s when everything disappeared. I know there are like 3 relics of it. And I can show you the tree actually, the leaves, you know.
Did you
on
say
the live
Hilliard
shoot.
Woods
Yes,
near Rutgers?
that's part of Rutgers Gardens, yeah.
Yeah, I wonder if Gene Varney and Ray Fatto maybe inventoried it. Maybe John Burkhart, do you maybe have lists from there?
Okay, so here, this is in the fall, so you can see some of the leaves on the live shoots, on the shoots that look healthy. And I collected a few leaves and I pressed them. This is for the Chrysler Herbarium, okay? Where I volunteer, okay?
Oh, wow.
Okay, let's go back to the fungus. Okay, so this is like on the dead branch, okay? The branch that appears dead. On the live branch, we can see a little bit of orange here. Unfortunately, I don't have a good picture.
That seems right, Hervé, because I've observed that on the same thing that you're saying, you know, because the roots are still alive and they send up the shoots and the tree lives for a few years until the chestnut blight, which is still just existing in our ecosystem, attacks it. And they're just like little orange dots that erupt out of the bark.
Yeah.
I've always seen it, I guess, in the later in the year, I think in the fall, but that doesn't mean anything. You know, maybe it just likes cool wet weather.
So so here are the early leaves in the early spring, okay? I just would like to have a confirmation that this is the chestnut blight that we see.
What's the name?
Sorry?
What's the name?
The name of what? Of the blight?
Yeah.
Okay. It's here. Here it is. Cryphonectria parasitica.
Oh,
It seems like a good start there, Hervé. I don't know if, I mean, I can't personally say from those photographs, but that is what that stuff looks like. Little orange dots that erupt out of chestnut.
Yeah, yeah.
Usually there's a lot of like splits and ruptures in the wood where that's happening.
I've been checking in on a lookalike for that one, the Melogramma gyrosa. I've been keeping tabs on one that looks a lot like that.
Oh, yeah, see those splits in there? See
Yeah,
how
I
it's
think
like,
yeah.
I would say you're, I mean, you probably, if you want to be 100% sure, you probably need to take a microscope to it, but I think you're pretty darn close there.
Yeah.
All right,
Okay.
cool.
All right. That's
Neat.
it.
All right, great. Thanks, Hervé.
That's my last observation. Okay.
All right, thanks for showing us.
Okay.
All right, how about Derek?
Hello, can everybody see my screen?
Yes, we can see your screen.
Okay, great.
It's really loud.
Oh, let me turn it down 1 second.
Thanks.
Is that better?
Yeah.
So I only have one mushroom to share today. This says wine cap. Somebody else identified it wine cap on iNaturalist already. And that's what I think it is, but this is my first time ever seeing it. So I just wanted to be sure. So they were growing in wood chips. These are what some of the younger ones look like. And then these are what some of the older ones look like. And I didn't notice any ring around the stem on any of them. I read that that was something that you would probably see if it was a wine cap, and then I did a spore print, and to me it looks blackish, but.
Purplish blackish. And that is correct, the identification, I would say.
Yeah, I'd say also. And it's always a good idea to take a sport print on both white and black medium, because the really dark print like this, when seen on white, is going to look like it's jet black. But
Yeah,
on a black background, you might be able to pick up the subtle tinge of purple brown that probably is part of the spore print. And this does look like Stropharia, and it looks like I can see remnants of the partial veil.
yeah, the younger ones definitely had the partial veil there.
Oh, well, yeah, okay. Yeah, you should get some pictures of the of like several undersides, right?
Okay.
So we can all see that. That's always a good idea. But yeah. So what happens is sometimes the partial veil sticks to the cap margin and tears apart instead of forming a ring, or it might get washed out by rain or something like that. But yeah, I mean, that certainly looks like wine cap Stropharia to me, and it's the right habitat and the right time of the year.
Okay, well, thank you very much. And that's a good idea about the spore print.
Are they still calling it Stropharia rugoso annulata? Used to be 3 words, but now it's just, yeah, okay, they've combined the rugoso annulata. Okay.
At least all the letters are still the same.
It's pretty tasty too. I ate some last night.
Do you eat the stems?
Yes.
The
Yes,
stems get a little bit tough when they get older, but I guess when they're young, they're good.
just what you have pictured here is how you want to pick them for eating.
Yeah.
Yeah.
And I don't remember about bugs in them, but as they get older, they get a little more mushy, I think. But they can grow huge, big. And if they're still in good shape, they're very good eating.
All right, well, thank you. Good to know.
All right, thanks, Derek. All right, anybody else want to share their screen before I go into the emails? All right, then.
Look, I have a question on the wine caps. Can the mushroom caps be concave, or are they always that arching?
No, when they get older, they definitely become concave. In fact, I think I have a picture of one where they're like that.
The other thing they do is they fade in the sun a lot. So they're not always that beautiful wine color. They get bleached out by the sun. And there is another species, I forget what it is, that sometimes looks like it.
F. luteum. Stropharia rugosa annulata, F. luteum. Apparently it has the same DNA signature, but it has a yellow cap. I find it here in Pennsylvania occasionally.
Who's burning shit down here?
And the other things that might be confused with Stropharia, especially in the spring, are perhaps species of Agrocybie, which have more or less cigar brown or at least brown sport.
See, there's a picture of them when they're a little bit older. With the cap upturned. Is that what you were asking?
Yeah, that's what I saw today too in my garden on the wood chips. I just didn't get a chance to put it into iNaturalist or anything.
Yeah, so like these I didn't bother with because they were a little too far gone to eat. They're like all gills. But that one is good edible stuff. For like that, these little tiny buttons.
Yeah, see, the color is what throws you initially because it's not a wine color. But
Yeah,
yeah,
you're right. Like that one's a little redder, but yeah, sometimes they are yellow.
or beigey, bleached out like. But the other characters, and the root at the bottom that often goes into the wood chips, that's helpful.
Yeah, right.
Yeah, the rhizomorphs, the white rhizomorphs. That's a Stropharia trait. But I think Agrocybie also has those sometimes too. Some types of Agrocybie.
And you'll see the Agrocybie and these things growing right by each other.
Yep, I found them together. Yep, a few feet apart.
Okay, thank you.
Sure. Okay, so in here, in the order of receipt, Marisol was the first person. You ready to go, Marisol?
Yes.
Wow, that's a mouthful. Pachycytospora.
Right. So on Saturday, I went to Glassboro, where my daughter lives, and we went to a park called something like Winona Park. Something with Winona, I don't really know. And there is a lake, Winona. So we walked. The park was very narrow, but it was in the side of like on the hillside, kind of. And it has mature beech and oak trees. And as we were walking, when I saw a gigantic dead, maybe oak tree, and it was hollow. And this thing is growing on the, whatever part is left of the thickness of the tree, in the right side is the hollow part of it. And it's growing inside too. This huge resupinate polypore was growing. You can see in relation to my hand. And it was beautiful. It has these drops of like wine color. And you can see that it has this kind of color on it, and it's thick. I measured the pores and it's like 1 per millimeter. And I put it down to get the spore print, and I could not even get one spore. And when I did the micro, not even one basidium. So I don't know what's going on with this, if it is still immature or what. But the micro characters are gorgeous. I found all sort of cystidia and gliocystidia and oh, and the whole crust is covered, is velutinous. I don't know if that's the right way to say, like very fine hairs, which are some of the exerted microstructures, yeah, covering everywhere. And when you touch it, it becomes like jelly.
Where did you get this name from?
Somebody on the cross fungi and polypore group in Facebook posted something like this a week ago.
Ah,
From Carolina, somewhere there, or Maryland, somewhere there. And I found mine and it looks like the same. And the same mutation, the same drops, the same aspect, the same color.
kind of like a, is that purplish color? That's not bruising, is that not?
No, it's untouched. Right there it is untouched. And it's really thick. And it grows like, you can see layers of growth.
Yeah.
Like concentric, almost you could say. Yeah. But I don't know why I could not get any spore at all. But it presents a really weird aspect. It's very irregular. And somebody suggested that name, but we don't know too much about it. Nobody knows. Nobody made any comment. Only that man who found it in Georgia or Maryland, somewhere there. So those are the structures that are sticking up like hairs, almost like. And there is Gloeocystidia. It's like imagine like a long sausage kind of thing with color, orange, yellowish color. And the contents came up very easily. Can you show more? Yeah. And you can see some septa at the center of the picture. And some thick walls too, you can see that. And those are all, these were super huge structures. There were so many different structures. I never saw anything like that, and I can't find anything like that. No idea.
Maricel?
Yeah?
I just punched in that genus name into Mushroom Observer, and there's a single observation of Pachychaetospora, and it's called tuberculosa. And that observation actually comes from Europe, from Germany.
Okay.
So it looks kind of similar. I mean, not necessarily the same species, but you know, they could be just related. You know, all of them kind of look the same, but the same mode of growth. You know, on oak, on the bark, or on the inside part of the hollow.
Tuberculosa?
Tuberculosa. Yes.
Okay. Thanks.
Yeah, I'm looking, most of the photographs online of this genus is that species, tuberculosa. And it does look pretty similar.
Oh, okay.
Kind of purplish.
I'll check that, tuberculosa.
So maybe it's just circumboreal or whatever you want to call that region.
Oh, yeah.
You know, just below the boreal. You know, it's no Nearctic and Polyarctic or whatever. You know, so.
Yeah, pretty neat. Never saw anything like that before, and it was gigantic. That was the impressive thing about it. No, the whole thing about it is amazing.
So it's just a decomposer, or this is a parasite?
This tree has been dead, who knows? Oh, this is the funny thing. The tree fell down, who knows when, across the path. So they cut it, but this side towards the base was hollow. The other side wasn't hollow. It was solid. Weird. But it was really huge, an old tree.
You know, when you're looking at these pores here, they look pretty immature. See how thick those disciplements are?
Yeah. And they are all covered with these hairs like things. You touch it and immediately it becomes gelatinous.
Neat. Very cool find.
Yep.
Yeah, Wikipedia says it's found in Europe, Asia, and North America. So.
Oh, the tuberculosa?
Exclusively on oak in Europe.
Oh, okay. Awesome.
So, yep, tuberculosa.
Nice. All right. In the same park, I found this polypore that I thought it looks like Polyporus arcularius, because I don't know, I couldn't find anything that looks closer than Polyporus arcularius, but I doubt it because it was so huge. It was so fat. And the only thing that didn't match arcularius was the size of the spores. The size of the spores of these things of this one are huge. So I put it on the Facebook group, Cross Fungi and Polypores, and 3 people suggested that it was Polyporus, the one that grows from tuberosus. So I read about it, and this can grow on the ground with the sclerotia, there is like a giant potato, or on wood, on deciduous wood. So this one will be the one growing on deciduous wood.
It isn't Polyporus radicatus?
No, I don't know that
With
one.
a deep root. It looked to me like it should have a root that went down into the stump of the wood and it got kind of cut off. But I remember something like that in New Jersey years ago with, see how tiny the pores are in the shape of it? But roots down and
On the ground or on the wood?
well, on the ground, in the ground, but it has to be very well rotted wood.
No, this still have some shape, the wood, even though that it was kind of old, but it still was.
But it doesn't look like he got the bottom is kind of part of my point.
I don't know because I didn't know what it was. So there were 2 together and I didn't want to damage the other one. So the other one was attached there. The other side is roundish. The other side of but I don't have the other side of this photo, but you know what I'm trying to say? It's like a cylinder.
Yeah, I find Polyporus radicatus occasionally. I usually don't find it until at least late summer or fall. It's usually has more color and the cap is flatter.
Hmm.
This doesn't really look like it to me, to be honest.
I'm sorry to interrupt. I'm looking at the mushroom observer observations of Polyporus radicatus, and all of them, as Dave said, do come from late summer into fall. It's not a spring species.
Oh, okay. And I have the book from Bernicia, Ana Rosa Bernicia, about the polypores, the last one from last year, and all the micro characters correspond. All these different kind of cystidia and the clams and these huge clams, it corresponds, plus the size of the spores is humongous. I got all the measurements in the observation.
What did you call it? Tuberaster?
Tuberaster. Mm-hmm.
Tuberaster, yeah. There's another one out west there. I don't know if it's the same species. They're using a name out in California now, Polyporus McMurphyi.
Oh.
But that might just be a Western one. But it is a name that is applied to what they call Tuberaster out there. So.
I see the version in the West. Mm-hmm.
Yeah, well, you just might want to look into that name and see if it's.
What's the name?
Maybe if that's a name that now applies more than just out west. McMurphyi.
Murphy. Okay.
Yeah.
PH.
Like the guy in One Flew Over the Cuckoo's Nest.
Is that PH or an F? M-U-R?
U-R. I think it might be double R. I'm not sure.
Oh. I'll find it.
I think it's PH.
I
think it's YI on the end.
Okay.
Yeah, it might be the new name for Polyporus tuberaster in North America. I'm not sure. You just might want to check that out.
Okay, because somebody suggested also Bresadolia chanterella.
Yeah, I don't know.
Bresadolia chanterella. I've never heard about it, but the spores didn't match. So yeah.
Bresadola craterellus.
Craterellus. Yeah.
No, it's not craterellus.
No, I didn't agree with that one. And plus the spores didn't match the size. We're smaller than this one. And it was funny because I also find another Polyporus squamosus, Seriporius squamosus, and the spores are identical except that they are smaller. And I have that observation is the next. I thought I was like, when I saw the spores of the next observation, I was like, what? And then when I measured them, the size is smaller, but the spores were exactly the same idea. In the next.
The next observation? Oh, okay. I see what you're saying.
Mhm. It was smaller and single, and it has a little bit of black foot with some kind of scales. And the pores are a little bigger. Right. And I did the micro of it. Oh, one cool thing about the observation before this one, the one that I call polyporous tuberaster, you touch it and it becomes gelatinous. It was a weird thing. I don't know why. Very delicate. Okay, so I did the micro, I have the spores for this one too. It's exactly the same idea.
All right. You all done with that one, Maricel?
I'm done here. I didn't have too much to say. In the same park, I found this Ganoderma applanatum, and there were like several fruiting bodies. I took the photo of two, and one was like a bell. It was attached just by a central point. You're going to see photos of that one, not this one, the next. That one. So when I took it off the tree, I damaged it, and it has 3 layers. So I remember reading that Ganoderma applanatum is perennial. So here the white part is the new growth, the new pore surface growth. The one that is ochre creamy, that's probably from last year.
Well, that's interesting to see that it grows from the center outward.
The way it was attached to the wood, it was attached like a wasp's nest from one central point. The other ones were attached wider.
Okay, so that
You know what I'm saying? Yeah.
That might just be
Right there. That's the one.
That might be something that's just particular to this fruit body.
Yeah.
Yeah.
And it was hard to detach. The top part was hard to detach.
That's a really good photo there, showing the new growth spreading out over the old pores.
Yep.
Is the staining of the pore surface consistent with the artist's conch?
It is the artist's conch if it's Ganoderma applanatum.
Yeah, it is, right. Okay. So we established identity of this mushroom pretty well.
Yeah. It has the best white pore surface usually later in the summer, but they start to come to life now. Obviously where you are now, but for me, some of the perennial polypores are starting to come to life. You can feel underneath, they're all putting out a new layer of pores. For as long as they have food from whatever they're attached to, they continue to grow bigger.
Yeah, there were so many big trees, dead big trees, deciduous in this area. Oaks and nuts and beech.
Right. Yeah, this would be considered the artist's conch, which is very common up here for that craft.
There's a really good one in the museum where you work, Susan. I'm sure you know that.
Oh, yeah.
There's a really nice big one. With
there's a lot of them around up here. In the Wild Center, it has several that have by well-known artists. And I actually have, this is how I got into mushrooms, is with Ganoderma applanatum when I was about 8 years old. Of course, we didn't call it that, but I spent, when my family would come up here to the Adirondacks, about an hour south of where I now live, I learned to identify that mushroom in the woods so that I could draw on it. Because the other thing with that, Maricel, is if you had picked that and taken it home and left it out to dry, good air circulation, not necessarily in a food dehydrator, that's too quick. And also being thick, it would take too long. But anyway, you can keep that forever. It'll dry and you'll be able to keep it. The trick, bugs will sometimes attack it later. But
I have a few Ganoderma. I have many polypores. I have a collection of polypores. And what I do is that I put them at the foot of my refrigerator. So the hot air comes through there and I leave them like for a week and I keep checking until they are very light.
Right. As long as you don't touch it, you can keep that permanently.
Oh, I see.
I can't quite see what the date is, but I think it's
2014.
Yeah. So that's one of my crude examples of what can be done up here.
Yeah, I have a collection. I dry them. Yeah. It takes like a week. And I keep turning them and it works.
The mistake is
to draw on it before you dry it though, right?
Huh?
You have to draw on it before you dry it. Is that true?
Yes, yes. Yeah, the real way to do it is, yes, while it's still, and I was just going to say, if you pull it off the tree or branch like this, you've got a beautiful imprint of your fingers. So you have to grab it off the tree like this and carry it out of the woods like this, so that, until you're ready to draw in it with a toothpick or something like that. And you usually have to do it within the same day you've picked it, because it start to dry out immediately.
But I got news. The Ganoderma expert, he uses alcohol with a paintbrush and he can draw. It reacts with the alcohol. He showed it to us.
Well, there's a lot of things you can do with it once it's dry, but you can paint on it. You can etch it with a wood burning Dremel, I forget what it's called. But yeah, that's a well-known art craft up here.
Yeah, good, thank you.
So here we have a Pisiza cup fungus.
Did I show this to you last week?
I don't think so.
No, okay. So this one was found in Franklin Parker Preserve near the parking lot and next to the edge of the brook and the bridge. That area is pretty special. So I was coming to the end of my walk that day when I saw 3 Pisizas on the ground. I say, what? And then it was very striking because it was white. You can see the little one towards the top, right? The little one. And
how large is it? How large is it?
It was, let me calculate, maybe 3 inches.
The big one?
Yes, 3 inches in diameter. Yeah, and it was incredible how white it was, and it was like brownish inside. And then it was growing on humus. That's how you say humus, the wood, the composed, powdery, deciduous wood. So it was growing there. And there is another one in the right, and it was completely brown inside and brown outside. And when I took it off the ground, it has a central point of attachment and it still had a little white. So I got a sample from the brown one and got this one, and they both have the same micro character. So it's the same one. So when it's very mature, it changes to brown. And I didn't know what it was because I never found one white one. And then Gary Taylor, he told me that it was Pisiza varia. So then I call it Pisiza varia.
Smooth spores?
The spores are not too ornamented. I got some pictures there. Not too ornamented.
Yeah.
But I see some double walls, and I was impressed by this donut-like aspect of several of them.
I think that's the cotton blue, which is kind of thick, collapses the wall.
Oh yes.
I think so.
But I never saw that in a Pisiza.
I also use Congo red. It's thinner.
Oh, I didn't know that. Can you
Well,
show more photos?
I don't really know it either, but I think
it produced that reaction. Huh.
It's cotton blue and Congo red, isn't it?
See, I use cotton blue for this one. I usually use cotton blue for the ascomycetes.
Right.
Yeah.
And I get these spores from a spore drop.
Yes, I left it throughout the night. I left both samples laying on the one
Yeah.
next, throughout the night, and the next day I got a good spore print.
Yeah, that's.
Yeah, that's good because with pizizas, if you squeeze immature spores out of an ascus, you might not get the true picture of the ornamentation.
Oh,
So
okay.
sometimes they're smooth when they're still in the ascus, and then they develop as they mature. They develop ornamentation. But
Oh, I
these
didn't
look
know
like,
that. Okay.
yeah. But if you got mature ones from a spore drop,
Yeah, I
you
did.
can be pretty sure that you're seeing the correct lack of ornamentation here. And
Oh, okay.
Pizizavaria, which, by the way, used to be called Pizziza repanda also. That's a
Oh, okay.
synonym. And I think the one we're seeing here probably would have been called Pizziza repanda maybe
Oh,
10
okay.
years ago.
All right, cool.
But they've been lumped together now, those names.
Cool. That was so cool that it was white. Yep. Outside.
Yeah, they're white on the outside. Sometimes they're yellow on the inside too.
Oh, okay.
Area repanda types. Sometimes they're really pale on the inside. Yeah, but they're usually kind of maybe not pure white on the outside, but pretty grainy too on the outside. Usually is what I seem to remember about these guys. I haven't found any yet.
Yeah, I haven't really seen very many Pizziza around here in Philly either. Sure. Usually you see a lot of them. All right, cool. Thanks, Marisol.
Okay.
I think it was your last one.
See, that's the last.
Yeah. All right. So who is next here? So.
Right. Now, two of these I showed you last week, but these are new groupings of them. And last week this is the Calocipha fulgens. The name was just up in the corner on the left. We talked about this coloring as part of the fruiting body, but I'm thinking maybe you guys are right that this might be a stain maybe from a fungus that's attacking this. I don't know. I haven't had time to really investigate it, but the way this one is kind of growing and collapsing and stuff, it really looks like it might be something that's parasitizing it. So anyway.
No, it's supposed to blue.
It's blue because it's supposed to be as part of the, it's not a stain.
I think it is staining in older age. That's what it says in a description of the species.
Now, see, when I usually see the blue-green when the young ones, and as they get older, they turn yellow. This week, there's a lot more of them around over at Scarface where I'm looking for my sarcosomas. So that's where I got this picture. But it's an interesting fungus because in the 12 years I've been here, this is really only about the third year that I've seen them. And it's always a wet spring, cold kind of thing.
Yep. Did you know that there are parasites?
The little cup fungi are parasites themselves?
No, this one. Apparently, it parasitizes the seeds of pines or conifers, I should say.
Oh, now that makes sense. That would be the seeds before the squirrels get them, huh?
Yeah, well, yeah, and it's a boreal mushroom. We don't find it in New Jersey or even, where they lives, or southern New York. I think it's mostly like a, circumboreal.
Yeah, that makes sense. Yeah.
Creature. Yeah.
I only see it where it's really heavily conifer. And like I said, I've only seen it about 3 times, but this year and a couple of years ago, there were just clusters of it everywhere at this time of year in the dense conifer. But anyway, it's a really cool little fungus because it's so drab in that part of the woods anyway. And the pine needles and the brown trail that you're on kind of thing in the tree growth.
Yeah, it's a pretty mushroom. If you look in the paper that I sent you and Dave on the ASCOs from those Boston guys, it has a unique, I think, phylogenetic position. It could be like one.
Oh, I do remember that. Yeah, I do remember seeing that. Yeah.
It's not like related closely to anything else. I mean, it's by itself on that tree. So,
Yeah, yeah. I do remember seeing it was a funny thing by itself.
and it's a parasite. You know, it doesn't attack trees. It just attacks the seeds
Uh-huh.
on the pine cones.
Huh, interesting.
Yeah, it is.
Okay, the next one, Luke, is, I forget what, oh, yeah, this I showed you maybe 2 weeks ago. But it's hard to see the little pores, but now when I see this on the tree, yeah, the further down is the one that's cut in half. Further down. Down, down. Down some more. Yep, there. Okay. You can see that the bug has entered in at the bottom hole, has eaten through the veil at the bottom, which covers this little polypore, and started to eat out the pore surface. And in that, if it's mature enough, which I think it was, because there was a lot of white dusty stuff, that's its strategy to get its spores out, because it's covered otherwise, is the insect comes in, and there's several different kinds of insects that come into this little thing and then carry off the spores to germinate somewhere else. And I have only seen it on red pines or Scotch pine up here. But it's an interesting little fungus, and it looks like popcorn on the tree when you, I usually see it when I'm driving, which is kind of not the way to do roadside identification, but it's a very interesting little fungus, and I had not realized that it comes out so early in the spring. Although up here, this is now kind of mid-spring.
I think we do find it in New Jersey, but it's not
Yes.
very common.
Yes, and when we used to go to the Pocono Environmental Center, we used to see it over there, because the little fruit buddies hang around for quite most of the summer. But there was a red pine forest over at the peak that always had this on it.
Yeah, it's interesting you mentioned peak, because I think the only time or times I've seen it was in the northern New Jersey.
Yeah, yeah. Well, it's definitely,
I get it here on Scotch pine.
yeah.
There's a Scotch pine
Usually-
little plantation not far from where I live, and it's usually on a dead tree, right?
Right, right, right.
Is it Cryptoporus? Is that the?
Cryptoporus vulvatus.
Right.
Yeah, crypto being like a mausoleum kind of crypt sort of thing, because it's closed.
Yep.
I guess that's why somebody named it that way.
Yeah,
It's fungus.
it grows only on conifers. I don't think it's obligatory.
Right.
Only on
right.
pines, I believe.
Yeah.
Yeah, it's pretty neat.
I've only ever seen it one time here in the Philadelphia area, and I think the area may have been planted. I think they were red pines,
Yeah, that sounds right.
like around a reservoir. It was like pines that were planted around a reservoir.
Mm-hmm. Mm-hmm.
Is there some sort of big polypore, bigger, harder, darker that grows on deciduous wood? I feel like I've seen that, but on deciduous.
Not with this veil that I've ever heard of.
Okay.
When I showed it to you last week, the bugs had not started. They were so young that the bug had not started to invade. And it's always where it goes in is close to where the attachment is to the tree, leaving most of the veil intact. Very interesting.
Now, Susan, is the veil persistent, or eventually it ruptures and the spores begin to drop out of the?
No, the veil is almost like a continuation. In fact, you can see that.
There.
No, I understand, but I'm saying as the fungus matures, does the veil rupture?
No.
And allows the spores to
Well,
drop?
no, it usually has to be eaten, have a hole eaten by the insect.
So that's how it reproduces. The insect actually has to drill into the hymenophore and carry the spores on its back.
Yeah, I think it's thought that the possibly not even on its back. You know, a lot of these beetles have there's little pockets on its exoskeleton for the spores. So when it eats here, the spores are actually becoming lodged in the exoskeleton, and then it moves to a new tree. And when it makes these holes here, digging into the tree, because they're bark beetles, I think.
Yeah, could be.
When they dig into here, the spores are deposited in the live trees.
Right, well, I use the expression metaphorically. Of course, they're all hairy and they have, the body parts, the chitin, you have seams in there or whatever for movement, and it gets trapped in there. So when they do burrow into the new tree or bark, they leave those spores right in the opening behind them. Right.
Yep. Yeah, I mean, some of these insects have actually evolved. It's not even the seams and stuff. It's actual pockets that are specifically for the spores. A lot of wasps are like that. The wasps have that, those little pockets. There's a name for that. I forget what it's called, but
On the one that's cut away, you could see that the insect had actually eaten the pores. So he would have been eating the pores as well as the spores
There.
with it. You see that?
Right.
I think he fell out when I cut it open, but they've already been working on these.
So, Luke, are you saying that the mushrooms sort of adapted to the insects having those cavities in their bodies specifically, for their spores to be trapped inside?
yes. Yep.
I didn't know that. That's very neat.
Yeah, a lot of wasps are like that because the wasp have those relationships with a fungus that they transport the fungus from tree to tree, and it's actually their larvae that are eating the fungus and the rotted wood. And then when the larvae pupate and emerge to go mate and drill into new trees, they carry the fungus with them so that their offspring will have something to eat as they grow.
Mm-hmm.
Interesting. Interesting.
Yeah, strange adaptation. I think Cryptoporus valvata is certainly very unique among polypores in that way.
Right. That's why it's pretty easy to identify.
Even I can do it.
Yeah, well, I'm just saying it's a good one. You can sound intelligent if you're a beginner. If you find one that, it looks like popcorn on a tree and you cut it in half and you see what I just showed you, you're pretty sure what you have. Now this I am not so sure of. I'm calling it Entoloma vernum, just because I'm sure it's going to be pink spored and it's got a little umbo in the cap and it's a spring Entoloma. I was surprised to see it where it was, but even so, I can't imagine that it's anything else. It's way too early. We haven't even got morels here. We've got false morels, but not even regular morels. And this thing was, this is also almost already over the hill. So I'm pretty sure it's Entoloma vernum.
What do you think, Dave?
Well, I think there's what constitutes the true Entoloma vernum is kind of unsettled. And
Right.
I think it's believed that there's several different things that have gone under that name. And I've even found Entoloma, Strictius, pretty early during morel season. Entomomas start up really early. I found some entomomas yesterday on our property, and they might even be the same species as this. They don't look all that different. But I find what I think to be several different species of Entoloma that come up pretty early. And I did find some years back, really early, like April 15th or something, that seemed to key out as the true Entoloma vernum. One of the problems with Entolomas is there's usually not a lot to go on. They don't have cystidia. The spores pretty much look all the same. Some of them might be a little longer than others, like the quotient might be a little bigger, the length to width. But Entolomas are really, really hard, and there's really a lot of species.
Yeah, that's why I probably should call this Entoloma vernum complex.
Yeah, that's what I would call it.
Yeah. Well, the fact we don't get gilled mushrooms for quite some time. So the fact that I saw this day before yesterday is kind of surprised me.
Tim Baroni told me that every spring he used to, I don't know if he still does it, but he's the big Entomoma researcher in North
Right.
America, that he used to go to the Adirondacks every spring. Now, I don't know what he meant by spring. Maybe he meant June. But he said that he used to go to the Adirondacks to look for Entomomas because there were really a lot of different species that were undocumented that grew in the Adirondacks. And I guess he would look like in wet areas.
Well, I can tell you of my own experience here in several places. There's usually in July when it's really wet, or if it is really wet, the leptonias, the little funny ones that he likes. I always say, damn, I wish Tim Baroni was here because I find 4 or 5 different, noticeably different species of leptonia all within the same week. So anyway, an interesting find, like I said, for here to have a gilled mushroom at all.
Yeah, and they're terrestrial too, these early Entomomas.
Yeah.
Sort of equally interesting for a very early mushroom.
Now, the thing that's very prevalent this week and now in full size are the Gyromitra esculentas. I didn't take a picture of one that's cut in half, but it should be chambered inside. This is looking much more typically what I expected Gyromitra esculenta to look like. This is the false morel, folks. You want to sort of recognize this so that you don't eat it. Looks a lot different than the true morel. Yeah, they often will grow in little bunches with the little thick stubby stems. You have to get down at eye level. This was lucky because it was on a hillside, so I didn't have to bend down so much. I could look up into it. And these 2 different places. Actually, these first 2 pictures were next to the building at the Paul Smith's Vic, and this is sort of the other end of the property towards the campus. And then the next picture was, this was the one over halfway to Lake Placid. So they're very common up here right now. We've had a lot of cold, wet weather. So just, you can see how much they vary in their shape, and the size is all the size of small fists usually when they really grow out. Interesting mushrooms.
Susan, I don't know much about Gyromitras, but how do you differentiate it, that species from other species in America?
Well, for one thing, up here, they're all pretty much, I think, the same, although I haven't seen it, what I would call a Gyromitra corfii.
Right.
Because that's usually a much lighter brown and a little bit different shape. But since I looked at the spores once about 10 years ago, and they're really quite interesting because they have these huge oil drops in them. Sometimes 3 oil drops, if I remember right. But I have not really, or shall I say, gone into deeper identification with a microscope and keys. But I know this kind of, in fact, when I've posted these on iNaturalist, before I even finish posting the 2 or 3 other things that I have, somebody's already
jumped in it, right?
Yeah, they've already looked at it. They've already, agreed or otherwise. And one of them is Michael Walt, which it's got to be Walt Sturgeon, I'm sure. So somebody's following Gyromitras.
Yeah, Michael Walt, who doesn't do any DNA or even microscopy.
Yeah,
But,
well, that's, yeah.
Those can be differentiated from Brunium and Corfii, usually by the fact that they have a thin, well-differentiated stalk. As you see here, Caroliniana, Brunia, and Corfii. The Brunia does have a stalk that's fairly well differentiated, but they're usually really thick, and it's almost like an extension of the cap almost. You see here on the esculentas, see how the stalks are all relatively thin and long and very much stalk-like. That's one thing you can use. And the that would be, I'd say, one of the main things. I'm not sure. I think Esculenta has spores that look like one of the other ones.
Interesting, this picture shows the variation within each of those groups, though.
Yeah, yeah, the morphological variation. This is good.
Yeah, yeah, no two of them actually look identical in their shape and convoluting.
Corfii and Brunia can be pretty darn similar. And
Yep,
really,
yep.
sometimes you have to look at spores to tell the difference between those two. And you mount the
Like I
spores
said,
in-
I have seen something that I would call Corfii, but I haven't seen anything that I would call Brunia.
Yeah, you
Up there, I wouldn't
need to
expect,
mount the spores in water. You need mature spores, and you need to mount them in water, because if you mount like in KOH, the KOH dissolves the ornamentation on one of them. I think it's Brunia, where it dissolves the
Ah.
ornamentation. So then they're really hard to tell apart because the spores look a lot alike it if you do that. Esculenta has spores that look like one of the other three, but I forget which one. They
Lucas,
all have oil drops in them.
Did you put this up?
Yeah, this is from false, what do they call that? False morels
Oh, Gary
demystified.
Gilbert.
Gary
Yeah.
Gilbert and Wesley. Yeah,
Yeah,
this is a
uh-huh,
Facebook
uh-huh.
thing.
But
I know Gary.
Yeah. And these guys, this is obviously their opinion, but they're saying that the Esculenta are edible with special preparation, and they call all these other ones edible.
I wouldn't eat any gyromitras.
There's a lot of people out there that are really pushing it with these things. I've never eaten any of them personally, but
Yeah, but I've always been told it's like a cumulative poison. Once your body gets to the point where you've eaten enough, it's going to say, nope, and can kill you.
that's what some of these guys like Gary Gilbert are pushing back at. I don't know. I don't really follow it that much because I'm not really that interested in eating them.
No.
well, it does look like Esculenta has a unique look about it, and can be morphologically distinct. At least it looks morphologically distinct, from the other four, mostly by its stem, which is more slender and has more color in it. Is that correct?
Yeah, this
It's
stem can
always
be
that
white on
yellowy
these.
beige.
It's a thinner stem. It's
They-
more
never
of a
white.
differentiation between cap and stem. The other ones, it's like the stem the cap is almost like an extension of the stem.
Yeah, it fuses into it. Yeah, I know what you mean. Yeah.
Yeah.
I have seen that, but not, well, probably with the Corfii.
Dave, I think you're finding quite a variety of Dryomytris in your neck of the woods. Have you ever found Esculenta in Pennsylvania?
Yeah, not too often though. And I haven't seen Esculenta now for at least 5 years, but I have seen it. And when I see it, I know what it is. And pretty much I know immediately, I know it's not Corfii or Brunia. Caroliniana is the one that I don't see around here.
Well, you can probably imagine I'm driving at biogeography, that certain species have a certain distribution range, and
Caroliniana is a southeastern species. I think you might be able to find it in New Jersey. Maybe
Right.
once in a while. Corfii is our most common one around here.
Yeah, I was gonna say Corfii is the only one I've ever identified positively in Philadelphia area.
For the 30 years that I was looking for morels in New Jersey, I only found any kind of Gyromitra, I think twice.
Oh, you're lucky, Susan, because I've been looking for over 10 years. I've never seen a Gyromitra in my life.
Well, that's why I say it's they're not common in New Jersey, but boy, if you want to see them, you could come up here.
That's interesting. Yeah, because
from
I
several
don't,
different places.
because southeastern Pennsylvania is the same, very, very uncommon. Like I'll go a couple years at a time without seeing a single one.
I
But it
found
is-
Brunia in New Jersey.
It
And
is
I-
worth knowing that there are false morels, though. That's the thing.
Yeah. I
Or-
found Brunia in New Jersey, and I positively identified it by spore ornamentation. But I was pretty certain what it was to begin with.
Mhm.
But the spores had the right ornamentation. I
What
think
part of New
Brunia
Jersey
is the one that has just like these really translucent, hard to see little nodes that are scattered around, mostly concentrated on the poles of the spores, but not in any really highly organized way. And Corfii I believe, is the one that has knobs on each end. There's like a knob at each pole, like a single knob at each pole. I think, you know what? I've got my ASCO book right here. I'll look. But it's sport ornamentation.
Is the book you're looking at the Bissett and Bug,
Yeah,
Michael Bug?
yeah, I've got it right here.
Yeah.
That site where I got that that chart from, they refer to gyrometers as true gyrometers, and they refer to all morels as false gyrometers.
Yeah, that must be Arlene's humor. Huh.
Okay, you want me to go into these?
Yeah, well, yeah, that's my dominant mushroom right now, just because I've had such a kick out of them. All right, you've been looking at this same little twosome for now probably the 4th or 5th week. But if I showed you the first week's one, the only way you would recognize the same little fruit bodies is by the pine cones on either side of it, because it has changed so drastically over the last 4 or 5 weeks. So now this is becoming more typical, the big one anyway, of how it's sort of starting to flatten out. And I suspect now that I could get spores out of this. I didn't bend over to try and blow on it because it was fairly wet still when I was there. But it just fascinates me how they change so and fill up with water and then change shape over from week to week.
Great photo. Mm.
Yeah, I get to, yeah, and that's another one. This is more typically what they look like. That's about 2.5 inches across, 2.5 inches deep. Still fairly dark in the center, as opposed as they mature, they not only flatten, but it becomes lighter brown. But the skin is so interesting because we've had a lot of really wet weather, so they tend to be really plump. Now this is one that got nibbled on by quite a lot, so it's kind of collapsing, but it's not going away. It's still there. It's still holding its water, so to speak. But it's just interesting to see how I've had a couple that I've watched over about the last 3 weeks, including one that I was bending over to take a picture and stuck my knee in it and kind of burst it. But the silly thing is still there and is still full of water. Not that it's going to make any spores. Now, this is another little threesome that I've showed you, I think, or I know I've put it on iNaturalist. And I mean, that big one is like 4 inches across. Oh, well, someone who's faster at typing can, Igor, you can put the name in. Sarcosoma globosum.
All right, I'll type it in.
It's a snowmelt thing, really. It's where they get their initial moisture from. It's an ascomycete, and it is a spruce associate, which is what I find it here. I knew that sort of before I read that.
It's not a spruce associate because it's a saprotrophic fungus. It just prefers conifer environment for some reason.
Well, or maybe it eats the cones or the spruce branches, or you see all of the detritus kind of duff, as I would call it, all of the little twigs and branches. So maybe it works on that kind of stuff. But they're a really neat little fungus. And let's see, last week in my site number 4, I counted 106. Yesterday, I counted 124 in the same area. And that's just one of the 4 sites that I go into. And this, okay, so Igor is interested in trying to get DNA from these to see if they are the same as what is in Northern Europe and Russia. So anyway, I collected a couple yesterday, 3 of them. And this picture is cut in half, just to show you the sort of gelatinous interior. It's kind of like cutting a grape in half, and yet it still kind of oozed out like it was kind of leaking. But I'm surprised at how well they dried. I had the dryer at a high temperature overnight, and they turned into little, almost potato chips like.
Good. Getting all the moisture out is crucial for DNA sequencing, because if there's any moisture left, DNA will
Um,
likely decompose.
I
or you
didn't
could have,
actually touch them, Igor, but because I didn't want to, compromise that. But I poked them with a metal tongs kind of thing, and they were. I was afraid I was going to break it. It was that dry. So
Good.
I did send them to you today, and I did send it priority only because that's the box that fit it. So
Oh, thank you.
Well, I don't worry about the postage because my donation to whatever. But this is the picture of two of the ones that I sent to you.
Excellent. Well,
The
it's
one on
gonna
the
wind
right
up-
was,
I'm sorry, go ahead. You first.
I was just going to say, the one on the right is the only one that I handled a little bit because it had already been detached from its growing spot, whereas the other one was, I actually had to dig that out with the finger from well below the duff below where it was growing. But the little one was already detached. So I thought, well, that's perfectly ready to go. And I'm not, in the case of the little one and this medium one, I don't know if they were truly mature, whereas the third one, which is not pictured here, it might be mature enough if you're looking for spores for one of the halves of it. But I mean, if it's anybody, except for the Bulgaria or Rufa, what's the other name for that?
Galiella.
Galliella.
Yeah, that was the first name. That has a similar consistency inside, but in that case, as I remember, the disc is light brown and the outer part is the black. If I can't remember. I haven't seen that in years. I used to see that all the time in New Jersey.
Yeah, you
Galiella is
find that
yellow.
one in deciduous woods.
Right. And very common in New Jersey. Okay, I think that's all I have, Luke.
All right. Thanks, Susan.
Yeah, so this will be hopefully sequenced eventually, and it will go into our herbarium. I have no reason to keep it in my own, so we'll probably go to NJMA.
Sounds good to me.
Yep. So
I'm interested in the results.
Yeah, I'm too, because it could maybe show a phylogeographic pattern in its DNA, which is going to be interesting to see. And you should keep that spot secret, Susan, because apparently the species is not common and it's protected in Europe, at least some countries.
Yeah, I've been curious on the iNaturalist thing because on my when I look at my postings, it shows the map of where it is. Whereas there was something, if one of you pokes, in fact, Igor, you should poke into my observations on iNaturalist just to see, does it show the location? Because there was something that made me think that the location was being hidden from other viewers because it was listed as rare.
Yeah, you should hide it at any
Well,
rate.
I don't know how to do that on my posting.
Yeah, I'm not a INET fan. I don't go there.
Yeah, I know you said that, but I'm just
Susan,
curious.
when you're posting it, there's an option that'll say for location, it'll say open or obscured, and you have to click obscured. The default is open.
Okay.
So if you click obscured, it'll show up as a random point somewhere in the county that you're in.
Oh,
So it'll only show off the county that it was observed.
okay. I have not investigated that enough. And now that I've probably done 30 postings of it, if it shows where it is, it's kind of the secret's out, but you'd have to be really looking for them. They're very rarely right on the path. I have to, I mean, and you're not, they're this big mostly. You have to sort of really treasure hunt for them. This is a cool picture. Is Dorothy?
So this is Dorothy. I don't think Dorothy's with us tonight. She sent this to me. Are you here, Dorothy? I don't think she is. So these are Psilocybe ovoideocystadiata.
Wow.
They call them ovoids for short. So it looks like she found them on Saturday. This was just described in 2003, she points out. It was actually described right here in Pennsylvania, right? One county over from Philadelphia by a local guy named Richard Gaines. He doesn't live around here anymore, but I heard his story. He was saying that he kept finding these and he knew they were psilocybes and kept sending them down to Dr. Guzman in Mexico. And Guzman kept saying that they were cerupeles, I guess not really looking at them, or cerupeles. And this guy Gaines kept persisting, saying that they were something different. And finally, Guzman actually looked at them a little more closely, and it was like, "Whoa, you're right. These are different species." And he described them and called them this Ovoideo stadiata. And he put that guy Richard Gaines as a co-author on the paper. So just goes to show, keep looking hard and sending them into the experts, right? So Dorothy said that it has rhomboid spores and it fruits earlier than cerupeles, which you find more like in the summertime. And she says she didn't collect them because there were bird watchers around.
Oh,
And I guess the bird watchers would frown upon collecting magic mushrooms, if they even knew. So these are hallucinogenic. They're quite popular for collecting in this area amongst the hallucinogen crowd. And you can see the bluing on there, which I don't really know why these things blue. It's the psilocybin or the psilocybin in them, but I don't understand the chemistry. But they're not blue like that when you see them. They're pure white, but when you handle them, they turn blue like this.
When young, there's a partial veil. And I believe that that's a
Yeah.
difference between these and cerulees.
Yeah, and isn't cerulees, they're usually like smaller and daintier, aren't they?
They're smaller.
Yeah, these things are
You don't see them
pretty,
until late July.
yeah, these things are pretty robust. They're good, you know, I mean,
They're big.
3-4 inches tall.
They're big.
There's the veil or the remnants of it.
Mm-hmm.
They
I don't
like-
think cerulees
Fabulous
has a veil.
picture.
What'd you say, Dave?
I think cerulees has no veil. I'd have to double check on
Okay.
that.
Yeah, and the cerulees is more usually like leaf litter. And this looks like leaf litter, but these things are more like rotting wood, like wood chips,
Yeah, woody debris too.
like on the floodplains.
They show up in a lot of places in May. Sometimes all year, sometimes right through the summer though. I've seen them. There's a place 2 miles from my house where I saw them pretty much every month up until about mid-September.
We've
One
gotten them on the forays at Institute Woods, and we also got them when we went over, Luke, over at Pennsylvania, where you know, near Phillips there, that walk.
Yeah, yeah, I remember that. Right on the floodplain, or like right on the road that was along the creek. It looked like it flooded up there. All right, cool. Well, that's a nice picture.
I don't know about you guys, you know, collecting New Jersey. I've never seen psilocybe in our state or ever.
So apparently with these, if you know how to look for them, they're really, really common.
Well, if there are spring mushrooms, then that probably explains why, because I don't really look for things in the spring. Not even morels anymore.
These were gotten pretty close to the parking lot in Institute Woods on a foray several years ago. In fact, it was yeah. So they were found several times.
And I remember seeing in our herbarium, the NJMA herbarium, a
Uh-huh.
psilocybe specimen from Institute Woods from the 80s. And all it said was psilocybe species on it, because obviously this wasn't described until 2003. So whoever collected it back then couldn't put a name on it because there was no name. But I bet it was the same thing.
Oh.
You know, considering the same location and the same time of the year.
Right.
Are they more seasonal, or they are kind of spread out throughout the year?
There's definitely a big peak season right now.
May, yeah,
Yeah.
May. Usually when there's warm weather in May, like warm or hot weather, we'll bring them out.
If you're on Facebook on any of the local mushroom things, you can see that people are finding them in droves right now.
I see.
I think they're pretty popular for, collectors.
So you've found, you guys have had very wet weather recently?
Not really. Yeah, actually, it's been one of the driest springs, in my memory in the last few years.
Oh,
Had a little bit of rain last week, but not tons. Yeah, I think it's pretty dry too.
We had rain, but both in April and May, it's been below average. I think we might have gotten only 50% of our normal. And there were no big storms, just maybe like a 0.1 inches there, quarter inch there, that kind of thing.
It's a lot drier where you are, Igor, down in Pine Barrens and stuff than it has been further north.
Well, I mean, I look at the map of precipitation. Stokes is the driest spot right now in the whole state, and coastal Atlantic County, that's the wettest spot. But that's basically like year to date.
All right, Nina, are you here? Nina Edwards.
I'm here. Can you hear me?
Yeah, hi Nina.
How you doing?
Thank you. So here's your mushroom.
Thank you for including me. Yeah, I'm pretty new to all these mushroom stuff, so I'm just learning. And I captured this one in my garden the other day. And I went back today because I wanted to get a picture of the underside. So I tried to figure out which one it was by looking in some mushroom field guides that I got from the library, but I couldn't really come up with anything. Just I found the striations on the top cap kind of interesting, but I didn't know what it was.
Okay, well, there's the top. And here's the obligatory
those
underside.
Aren't striations, those are cracks or fissures. Striations are well organized radial grooves, usually more prominent along the margin. These are cracks that develop because the cuticle dries out. This is probably Agricybie.
Yep.
Maybe Agricybie, one of the species in Praecox group.
or Agricybie for some of us.
Agricybie, yeah, Agricybie, right? Yeah. Agricybie is probably the right way to say it.
That's kind of what I came up with in one of the books that was showing.
You get a spore print?
No, I didn't. I've never done that before. But I'm listening to you guys, so I just would get a piece of white paper and black paper and let it sit there for an overnight or something.
Try to use paper that's not absorbent. Get some like glossy paper. Absorbent paper will draw moisture out of the mushroom, and when the moisture gets into the paper, it stains the paper, and you might not get a good read on the spore color.
So I could use like freezer paper.
Yeah, that would be good. But try to get really white or really black, so you can tell any subtle differences. These Agricybie, though, they're usually pretty cigar brown. They're pretty unmistakably brown spore prints usually. So
it also looks
black or white.
It also looks like maybe your garden has wood chips or some kind of mulch.
It is mulch. Yeah, it does in a certain portion of it.
Yeah, that's a typical habitat for Agricibies.
Wow.
Yeah, and whether or not, if you find some young ones, observe whether they have partial veil or not. There are species with partial veil and species without partial veil that
What does the partial veil mean?
Oh, covering over the gills, the underside, like almost like a little bit of a membrane for Agricybie.
Yeah.
Other partial veils might be stringy, but Agricybie will have a really, really thin sort of membrane, which might form a ring on the stalk when it breaks apart.
Yeah, it looks like this one has a little bit of some material there.
Looks like it's got a little bit of something on the stalk, so it's probably Praecox. one of the praecock species. There's a few of them.
So are these poisonous? Just out of curiosity, I'm not going to go eat it.
I don't think I don't think anybody's ever got poisoned from them, but I think people who have eaten them say they don't taste very good. So
Yeah, that's what I always hear too. That
Yeah.
you could eat them, but they're not well regarded.
Okay. Cool.
All right. Well, thanks, Nina.
All right,
Thanks for
thank
sending
you.
that over.
If you're not sure what it is, you should not be thinking of eating it.
No, I wasn't going to eat that one.
No, I'm just saying, just generally.
when in doubt, throw it out or save it for study.
Yeah,
True. Thank you, guys.
sure thing. Thanks. All right, Dave. Got some observations of yours.
Oh, okay. I found one of these. Well, a couple weeks ago, we found one on a local club foray here. Just one really small one. Probably the same log. A fair number of them came out. They're really cool looking mushrooms. The Cyanophylla species name basically is for the blue gills, I imagine. And so they're a pretty cool looking mushroom. And I think they used to be considered to be like a hygrocebi or something, but now there's this genus Chromosera or Chromosera. I'm not really sure how to say it, but yeah, they're a pretty cool mushroom to find. Usually, it's spring. I don't think I've ever found these any other time besides spring. They grow on dead wood, well rotted wood. I think pretty much, if my memory serves me right, I've only found these on conifer. This group was on a hemlock log, and they have spores. Sometimes they'll call this shape pip shape. They're kind of like seeds, kind of shaped like seeds, and with a point on one end. I guess that's where it would have been attached to the basidium.
Ridiculous.
That's a cool mushroom to find. They're pretty small, but they're kind of showy, especially once you turn them upside down and see the gills. They're very slippery, like Gliophorus. If you know Gliophorus, which is related to hygrocebi, you can hardly pick them up. They're so slippery. When they're young, I guess they dry out a little bit in age, but yeah, they're a pretty cool thing to find.
I've found them at least twice up here, but they're not usually so pink. They're
I
usually
think you have to get them when they're young.
yellow on top, but kind of purpley pink underneath.
There's another species, Lilacina or something. It's Champignon du Quebec has it. They've got the two species. The other one has bigger spores. The spores are like over 10 microns, I think, or maybe about 9 or 10 microns average. And they're more yellow. And they look a little, the picture on Champignon du Quebec looks a fair amount different than these. These have really close gills for one. This is pretty cool. This Cudoniella clavis. It's a tiny asco that grows in really wet environments. And when the water is high after a rain, they'll be right in the water like these guys were. Well, they're in the water, but they're really attached to like vegetable matter, woody debris, little twigs, branches, grassy stuff. So I got spores to drop out of these. I didn't section or anything like that. But I got spores to drop, and they very well matched the description for Cudoniella clavis in the Ascomycete Fungi of North America field guide. So I preserve these. If anybody's really interested, I have them. They're very small, which we will see coming up in a subsequent photo. There you go. Look at that big one there. It's like about half the size of a penny. So pretty cool thing to find though. they apparently like really clean, clear water. I found these before. I found them last year, actually, in the same spot. It's where a spring comes right out of the ground. So it's very clear, cool water. And there's the spores, and the spores are described as um, being more or less elliptic, but sort of elongated. And many of them will be a little thicker on one end. So that's a distinguishing feature. And the sizes I got here, I think the Bissett Boyd book says up to 17 microns long. I think the biggest one I found was maybe about 15 microns, but the spores, that is. But yeah, that's them. They're pretty cool. It's a fun thing to find. And we got some decent pictures. So they're on Mushroom Observer now.
That's cool.
Yeah, so we were talking about, I think the next thing is is it the Pisiza or the Antiloma? I forget. Oh, Antiloma. So there you go, Susan. Found actually, Karen, my wife, found these in our yard yesterday, and then I went out and got some pictures of them. So I'm not sure I would call these Verna. I think I have found Verna before, and Verna's, it's hard to say why they're different. These have the striate margin. Now, I think Nina just mentioned before about striations. So the Agrasabe we were talking about before, that was a cracked cap. But if you look, if you zoom this picture right here, we'll see striations. These are very, very fine striations. So see along the margin here, see how those lines are all pointing away from the center. So this is marginally striate, you would say, about this cap. And they're definitely Antiloma. Antilomas are not stingy with their spores. You can usually get, I got spores I had a visible spore print in about 40 minutes off of this one right here. This is the one I got the spore print off. They're growing in a group. So it's a little group. There were like 5 or 6 of them together. The gills are really, really, they look almost free. If you could do a zoom on this one, Luke, we'll see here that these gills are very likely, I didn't dig in there because once you dig in there, you tear the gills apart from the stalk anyway. But I imagine you can just see barely a tiny little attachment here and there.
Yeah, right there.
So this is sinuate attachment. And sinuate attachment can look very much like free gills that are free. So these Antilomas in particular don't look a whole lot like Pluteus mushrooms, but some of the bigger, more robust Antilomas do look like Pluteus. And Pluteus sometimes grows on the ground from buried wood. So people eat Pluteus, and if you want to eat Pluteus, it's a good idea to have a little bit of knowledge of Antilomas. And if you have a microscope and you have any doubt, you look at the spores. Antilomas, like I said, they almost always give up spores readily, and you get what they call angular spores. So they're not smooth. The at the outside is not a smooth contour. It's got all these angles, and so those are definitely Antiloma spores. That's one way to know you have an Antiloma. Antiloma spores could possibly be confused with Anasabe spores, which are, they're not so much angular as nodulose or what they call stellate. But anyway, and then I'm not even sure what this is. I just smash mounted some stuff, and it's something in the gill context, I guess. And I honestly still have a lot to learn about interpreting a lot of these microscopic features, but this picture came up pretty good, so I put it on Mushroom Observer, and who knows, maybe somebody will tell me what it is. But that's my Antilomas from the yard, and sometimes they come out even earlier than this. Like I said, one year here on our property, I found Antilomas in mid-April. It was a particularly warm early spring that year. Also, last week we were talking about getting spores. out of Piziza. Oh, it doesn't say Piziza. It says Phylloscypha. This is a new genus. I don't know much about it, just erected. I'm not sure how many species are in this Phylloscypha. This thing used to be Piziza phylogena, and before that, it was Piziza batio-confusa, because it looks like Piziza batia, which is a later occurring cup fungus, usually comes out in August. And in my experience, it's not nearly as common as these things. These are out in droves right now. I'm seeing them pretty much all over the place. I just picked a few more today from some wood chips near my office where I work. Now, the spores are really what give these away, because there's really a lot of pizizas that you can confuse with one another. But these guys have spores that are ornamented. They're sort of wrinkled, or they call it varicose, or maybe little warts, but the spores are definitely ornamented. You can see, especially on this one that's horizontally aligned, especially on the right, you can see the pole has like a thickened a thickened wall. So also these are bigutulate, which means you see that there's two relatively large, they're not real big oil droplets, but they're big enough to see. There's probably a few other really tiny ones in there. But these spores and the dimensions of these spores, which are pretty much 20 by 10 microns, at least that's close. Everything matches Piziza phylogena. Piziza phylogena is a very common cup fungus that occurs in the spring. And also another thing, if you happen to have melters, which is really hard to get, the tips of the asci are amyloid. So this is mounted in melters. It's just a little bit of hymenium that I scraped off or cut off and mounted in some melters, and you can see the tips are blue. And these blue tips, there's quite a bit of blue, meaning, it's a significant portion of the end of the ascus is blue. I think pretty much all Piziza species have asci that have amyloid tips, but sometimes it's just the cap, right? The very, very end of the ascus that turns blue. These guys seem to have a fair amount of amyloidity associated with the ends of the asci. So there's a lot of these out there now. They're sort of fun to scope because they have pretty prominent, they have prominent features. But I'll tell you what, these, as we discussed last week with getting spores out of these guys, a lot of these ascos are really stingy with their spores. You lay them out, and 3 days later, if you're lucky, you put a little mounting liquid on your slide and scope it, and you might find 6 spores. On the other hand, sometimes you lay them out, and an hour later, and the slide is white. So usually, though, these guys are kind of stingy with their spores because they shoot them all out all at once, and then you get them home, and they're done doing that, and then it takes a while to get more spores. But at any rate, that's my stuff for that. I had a few other things too, but I had some computer problems, and a lot of my other pictures are on a computer that is now in a shop. But anyway, that's my stuff for this week. Thanks.
All right, thanks, Dave. All right, I had a couple things out. I was able to get out for the weekend. I only put a few things on iNaturalist. One of them was this one, Gymnosporangium clavipes. So this I found growing on cedar, the red Atlantic cedar, or I'm sorry, eastern red cedar. So it's a little dried out. When it's wetter, this stuff kind of puffs up a little bit. But the trees in this area are just covered in this stuff right now. You see when I'm looking through the tree, I can just see it all over the branches everywhere. So this stuff, this is one of the hosts for it. And then the other host that it needs is something in the Rosaceae family. So like apples, quince trees. I always see it on, I think they're choke cherries, I think is what they are. I believe that's what they are. I'm not 100% sure that host, but they get like these little like spikes all over them. These like orange spikes later in the summer, in the middle of the summer. So one of the rust in that family was that family Pucciniaceae.
Isn't it hawthorn? On hawthorn, I think, and they go from one to the other.
Hawthorn's one of the alternate hosts. Yep.
Yeah.
And shad. I'm working on shad bush.
Shad bush.
Shad, what is it called? Service berry.
Service berries. Yeah, I think that may be what I see them on, or service berries. They get the actual berries themselves, like they get all those little orange spikes all over them.
Yeah.
Like in July or something, like a little later in the summer.
I'm very disappointed because I love to eat shad service berries, but you can never eat them because they got all those stupid things on them.
Yeah, well, this stuff is like this is in Bucks County, right in New Hope, right behind my work. There's a lot of these cedars, and they're just all covered in this stuff right now. And then you also see the that other one that's back there, the Gymnosperangium juniperi virginii, the octopus looking one, on the same tray. So
On the same tree?
on the same tree, yeah. Yeah.
Orange spaghetti coming out of the gall?
Yeah. Yeah, these things really are hosting a lot of stuff.
I've never seen as many as this year, actually.
Really?
One with spaghetti.
That's
Yeah,
interesting.
the stage that settles onto the fruit tree does damage to the fruit tree, at least the juniper, Ginny, whatever, got too many syllables really. That one can damage fruit trees.
Yeah, juniper.
Yeah.
So this is another one that I saw. Now it had been raining, so I wasn't 100% confident in this. I was calling Copernopsis variegata, scaly one. So it was fairly robust. These things were, a couple centimeters high by a couple centimeters wide. A little hardier than a lot of these inky caps, growing directly out of this wood. And it looks like it had a lot of scales on it before it rained. You see, like we're all in there.
Yeah, that's what I'd call that. And I think you're absolutely right. It's just that they would have been more completely uniformly covered in scales with just little spaces between the scales, but it looks like there was a lot of rain and it washed up. In fact, look below the mushrooms. That
Right
looks
here.
like scales that washed off.
Okay.
I think that might be scales that the veil that washed off. At least some of that stuff.
Yeah. All right, cool. I don't know if I had much else that made it on here. I'm in the process of getting my house ready for sale, and everything's a horrendous mess, and I don't really have anywhere to work on mushrooms right now. Like my desk and everything is just piled with boxes.
Where are you going?
Hopefully somewhere in like the Upper Bucks area.
Oh.
Not far, just out of the city a little bit. We'll see though. The housing market's so crazy. So then these Agroscides, and I didn't put a name on these either. I just didn't really. But at first I was thinking praecox, but they don't have a ring on them. Praecox has a ring.
Michael
No.
Close says there's one species of praecox that has no ring.
Really?
Yeah,
Hmm.
there is a species of Agrassi that comes out in the spring on wood chips. And depending upon who you ask, it's either Agrassi putaminum, I think it is, and the other one is Agrassi sororia. But these don't really, these look different. These look like more of a praecox type thing. And look at the one on the right, though, lower right. Looks like there might be a little bit of a deposit on the stalk. Sometimes the rings don't last very long, or that might just be spores. I'm not really sure. You see what I mean there?
Yeah, I think
Although-
it's spores, but
Yeah, although you know what, this longitudinally grooved stalk, this like vertically grooved stalk, that's something you see on that other one I mentioned, Agrassi putaminum. I think it might be. If it's not that, that's close.
okay.
I think there's differences in spore sizes. For I think putaminum might have somewhat larger spores. I'm not sure. I'd have to review that.
Okay. Cool. All right, I think that's it for me. That's actually it for everything that I had from people. Oh, so somebody asked at what point do they turn into black goo? I guess they were referring to those Copernellus. Those inky caps, they seem to, excuse me, they turn into the goo pretty quickly. They grow, and some of them within a day. So I imagine these.
Copernopsis turn quickly. Copernellus may not even deliquesce. Sometimes
No,
they.
Coprinopsis doesn't.
Yeah, that's Copernopsis. Those variegata, those are Copernopsis. Yeah, they will deliquesce usually completely and sometimes quickly. The Coprinellus, like Coprinellus mysaceus group, Coprinellus section mysacei is what it's being called on Mushroom Observer. Sometimes they don't even deliquesce. The gills just get really black and they might dry out before they even turn to a liquid. Depends on the weather with them. But the Coprinopsis are much more readily deliquescent.
What about Coprinopsis lagopus? It doesn't deliquesce at all.
Look,
Coprinopsis
yeah,
lagopus.
that one deliquesces pretty quickly, I think.
It doesn't.
Oh, it doesn't?
It doesn't. Coprinopsis doesn't do that. There are 4 groups within that family.
Oh, they don't?
No, they don't. I know it because I have seen it in front of my nose at work so many times, and I took so many photos of them, and they never did it. They just curl up and become like a bowl. And then you can see only the black lines on the gills, but they never turn dark or deliquesce.
They do become gooey and they drop some liquid. So that's their way of deliquescing, as opposed to a complete meltdown.
Okay.
And what about Coprinopsis ultramentaria and Coprinopsis romanesiana? Do they deliquesce?
Ultramentaria does. I think they're a little bit more slow, though.
Yeah, they're quite robust.
Yeah, they're big. They're thick fleshed. Yeah, you know what, I'd need to review that. Marisol might be right about that. But I thought especially the dainty Coprinopsis, I thought they don't even last very long. They'll come up and they'll be gone.
Yeah, they start in the night, and at noon, they are kind of done if it is too hot. They don't last too long. They bend. The stem breaks, and then you find them bending down, but they don't deliquesce. I mean, I observed them so many times. I may be wrong, but I think that I read about it.
You know,
That's why there are four groups within that family. The Coprinopsis, the Coprinellus, and Coprinus, whatever names. There are 4.
Yeah, Coprinus, Coprinellus, Coprinopsis, and Parasola.
Parasola and Parasola. Yeah.
All right.
I'll look into
If
that.
you want to read about it and tell us.
All right.
So we have about
I'll read up on that.
we have about 5 minutes left. I just want to check. Is there anybody that wanted to share that didn't get a chance?
I have a question for John and Nina Burkhardt about Cryptoporus volvatus. And the question is, did we ever find it in the Pine Barrens?
Yeah, I think we have. Cryptoporus volvatus.
Yeah, is that the,
Found it in Smithville. Sorry.
is that the thing covered up? Which one is that?
Yes, yes, that's the covered up one. Yes,
Yeah,
the one that
we found those, Marcel, we found those on the, across from Speedwell, haven't we, after the fire?
Oh, maybe.
Yeah, the things covering up the floors.
The pore.
Right.
No. That's Poronidulus. No. That's the tiniest polypore. This is a different one.
Oh, okay. No, then
No, that's the veil one.
Yeah.
The one with the veil.
It's the one I showed that looks like popcorn. It's about this
Oh, yeah.
Yellow.
Oh,
Yep.
that one. Oh, yeah. I don't know.
kind of a yellowy beige color.
Yeah, I'm
Igor,
just interested about its distribution range.
Igor, why are you asking?
Well, like I said, I'm interested in its distribution range. And
I found it. I found it
seeing
near me in Smithville
Okay, because I
on
only
a-
see it in
on a
northern Jersey.
standing conifer, the white pine tree,
Okay,
on a standing and dead.
I guess the answer to my question is going to be on Mushroom Observer because they have all the maps with pins, so I can same thing with INAT, I think.
Oh,
Yeah.
okay.
Okay, well, thank you.
All right.
Oh, Marisol,
Yeah.
I have David Aurora's book here.
Okay.
It used to be Coprinus legopus when they were all in Coprinus. So
I
anyway,
know,
he
yeah.
says, so we're probably sort of both right here. It depends on the weather. He says, we're deliquescing in wet weather. So maybe, when it's not wet, maybe they do just dry out and shrivel up.
If I see them again, man, I took so many photos of them. I was after them, but I read about it. That's weird. Okay.
Yeah. When I've seen them, and there's another one too. There's one or two other species that look like it, and the names escape me right now, but they're shiny, grayish, silvery Coprinopsis species.
See, there are a few other species. I just know Lagopus because it was in front of my nose.
Right. But
And the
I'm just
parking
saying,
lot.
they all seem to, I guess it requires the right atmospheric conditions, but they don't like turn into a ball of goo, like a Coprinus comatus would. But they kind of, the flesh gets very, very, very thin and blackish on the gills. And I've always thought of that as being a form of deliquescing, because they sort of turn into nothing in no time. But I don't know, maybe maybe in very dry weather, they just persist a little longer and just shrivel up.
The habitat where I found them is on the parking lot at my work. So it's on the lawn. So it's not really the woods. It's in the open. So there is nothing wet in there unless it rains.
Yeah, well, I don't really find them in the woods. I find them on like grassy debris. We had a bale of hay in the yard a few years ago, and a whole bunch of them came out on the bale of hay. May not have been Lagopus. There's another couple of species.
I can show some observations.
Cinereus is another one. That's very similar.
Coprinopsis cinereus?
Yes, okay. I say Cinereus.
Oh, I'm so sorry. I pronounced it in Spanish. Yeah.
Yeah, well, I know I might be saying it the wrong way.
No, but that's how I put it in my brain, cinereos. So I am saying it the way
Yeah,
it
well,
let it sound. Yeah.
you might be closer to correct than I am. But anyway, that's another one that's pretty similar. Anyway, I'll try to read up on that a little bit.
I'm
more.
going to show my observations of Lagopus next week.
All right.
Yeah.
I'll see if I can find any around here. It's been wet. It's supposed to warm up again. So maybe some will come out. We get them on our compost heap sometimes too, but it might be Cinereus. They're hard to tell apart really, Lagopus and Cinereus as far as I can see. And you try to get a spore print. And, oh, that's right. Yeah, you know what? When I try to get a spore print from these types, put the cap on the slide, and like an hour later, it's a puddle.
I ever find one, I'm going to try to do that, okay?
Yeah, see what happens. Try that out.
I'll tell you.
So Igor, back to the Cryptoporus question. I'm looking on
I was just looking it up myself here on Mushroom Observer. Go ahead, Luke.
I'll just say on iNaturalist, there's 3,238 observations of it.
Gosh.
And it's all across the Northern Hemisphere. Super heavily concentrated on the East Coast and the West Coast, all the way from Alaska down into Mexico and like up into Canada, down into Florida. But then you can also see like on the other side of the world, China, all over Europe.
Yeah, the veil for it.
That's strange because on the Mo, it's only in America, and you're right. It's concentrated on the western and eastern coasts, and the middle of the country is completely bereft of that species. But on the east coast, it only goes as far on the Mo as Georgia, and there's only one observation, I think, from New Jersey, or maybe actually none. It's in Philadelphia. I have to blow up the map and see. Nope, there's no observations from New Jersey of Cryptoporus alvatus.
Okay, but in iNaturalist, yes.
You guys got to get busy. Some of those maps can be a little bit not accurate. They tend to often be where there's folks like us.
Yeah, I'm looking at all these observations on Mushroom Observe. I mean on iNaturalist. So all these ones I'm looking at in Europe, not a single one that I've looked at so far was at all convincing to me. I see some Felinus in here.
Yeah, I wonder if Valvatus is a truly Nearctic. If I'm correct about the ecological realm name, it's North American species.
Right.
It's not really European or Eurasian.
Yeah, none of these Eurasian observations on iNaturalist look very convincing to me. But the ones in North America, I was doing some random ones down south and up north. They all look good.
Yeah, it's more common sort of toward the north and the east side, but on the west side they go all from Vancouver pretty much all the way down to Mexico, or I should say the Baja, peninsula.
Mm-hmm. Yeah, a lot of times there's like a lot of polypores when they first start growing too, and they're like babies. Like Ganoderma is a good one. They look like Cryptoporus if you don't look at them very carefully. That's what I'm seeing a lot of these observations. So.
Okay, so they're just misidentifications.
Yeah.
That's what they were.
So. Well, anyway, that brings us past 9 o'clock. So I'd like to thank everyone for participating, and I guess we do have a lecture this Friday coming up. Greg Mueller on I don't have it in front of me, but he's doing a lecture on conservation of fungi and all the things that go around that. So I hope everyone can tune into that, and then I will see everyone here next Tuesday, right?
Yep, thank you.
All right. Well, everyone have a great night. All right.
Okay. Bye-bye.
Good night.
All right. Bye-bye.
Bye-bye.
Bye.